Hey guys, in this video we're going to study mutations. I'm going to start sharing my screen with you now, and you'll be able to see our PowerPoint slides from chapter 10, and we're going to learn about mutations. So first of all, we're going to learn what does it mean when you say you have a mutation. Oops, trying to find my pen here so I can do some highlighting for you. So what's the definition of mutation? What happens to the DNA when it gets mutated? What's the difference between a point mutation and a frame shift mutation? What's the difference between silent, missense, and nonsense mutations? So we're going to have a lot of definitions to look at. So what's the definition of a mutation? A mutation is a permanent change in a DNA sequence. So that means that either the DNA polymerase made a mistake while copying DNA, or some other outside event caused the DNA to change. So we're going to talk in a later video about some of the common causes of mutations. So, but remember, if you change your DNA, you change your messenger RNA, and if you change your messenger RNA, you can change the protein that you form from that DNA. So sometimes the protein won't fold correctly and won't work correctly if there is a mutation. So another key thing about mutations is that they're completely random. We don't know why, we can have some guesses at why a mutation might occur, but we don't know where on the DNA molecule the change is going to be, and we don't know how many bases are going to change or what the extent of the mutation is going to be. So all mutations are random. So whether they're caused by something like X-rays or UV rays or chemicals in your food, we don't know where the DNA will be changed or if it will be even changed. So mutations are a completely random occurrence, and we're going to change our DNA. So remember though, if you change the DNA, you're changing the recipe, which is then going to change the protein that is made from that DNA. And some mutations are as small as a single base. So you could have CGA, and in the mutation you switch out that G and instead put another A in there, right? So that would be a point mutation because you're just changing one of the bases of the DNA sequence. So remember, after you've had your mutation, one of three things is going to occur. The protein won't work correctly. So this would mean that by changing the base, you change the amino acids. By changing the amino acids, the protein did not fold correctly. And remember, if you have the protein that doesn't have the right 3D shape, it won't work correctly. So that's one possible outcome. The second possible outcome is that the protein works better. So sometimes when you're talking about Darwin's ideas of survival of the fittest, and you know, that the creatures that are best fitted to the environment are going to create the most offspring and be the most successful as a species. A lot of those things that are allowing certain species to do better in certain environments are due to mutations in the DNA. So sometimes you can get a mutation that's going to change your protein so it works better, but this second one is very rare. Usually what happens is this first one, which is the protein changes and it won't work correctly. And then there's a third option, which is that the protein is not affected. So we'll see more about these three when we start learning about missense, silent, and nonsense mutations. So how do these mutations occur? So I've mentioned this kind of already in this video. So we've got a mistake in our DNA. So how did that mistake happen, or how did that extra base or wrong base get there? So it could have been due to an error in DNA replication. Remember, every time you copy your cell using the cell cycle and mitosis, you have to copy your DNA. So that means every time a cell copies, there's a possibility for some kind of error or mutation in that DNA. And when you think about it, think of the places that people get cancer. Skin cancer. Skin cells are constantly copying to repair and replace damaged cells. Digestive system cancers like stomach or colon cancer. Your digestive system has to constantly replace the lining of your digestive tubing. So again, those are cells that are copying all the time. So cells that copy a lot are the places that are going to get cancer, because cancer is an accumulation of several mutations that goes on to cause the tumor. So any cell that copies a lot has the possibility of accumulating mutations, and any cell that accumulates mutations could potentially turn cancerous. So remember, though, that DNA polymerase is supposed to be checking our DNA copying to make sure it's doing it right. So the DNA polymerase is pretty good at checking the DNA. So we only get a mutation or a mistake about 1 in every 1 billion bases. So mutations due to DNA replication are pretty rare, but remember, some cells like skin cells are copying so often that this starts to become significant when you think about the number of times in a lifetime that those cells would copy to repair and replace damaged tissue. Now, sometimes, like I said, the DNA polymerase fails to notice that there was a mistake in copying the DNA. So in that case, a mutation might slip through the net. And then the last one is the one that gets a lot of media attention, which is, can certain things in your environment cause DNA to mutate? So radiation from X-rays or the sun, that could cause DNA to mutate, or chemicals like all those weird chemicals that you find in your food, the E numbers, the additives, Splenda, right? You always read these scare articles like certain things could potentially give you cancers. So there's a lot of environmental factors that are being investigated that could potentially cause DNA mutation. So here you can see what a mutation would look like. So here's your original piece of DNA, and then here's the correct copy. So in the ideal situation, there'd be a purple base here in the area that's circled. But if you notice in the mutant down here, instead of a purple base, we have a red one. So we put in the wrong base in the wrong place. So now we have changed the DNA. And the next time this cell copies, this mutant copy of DNA is going to copy too. So it's going to, the mutation is going to spread into the next generation of daughter cells. So, like I said, a mutation occurs about once in every 1 billion bases, but so about once in every 1 million base pairs is how often a mutation actually occurs. But because the DNA polymerase proofreads this, that's how we get it down to 1 in 1 billion, right? So you see various numbers for this. So 1 in 1 billion is like after the DNA polymerase has corrected. This is like if DNA polymerase wasn't there, how many mutations would we have? Then the frequency of mutations would be higher, right? But because DNA polymerase is constantly proofreading our new strands of DNA and double checking our new strands of DNA, the mutations are rarer than you would expect. So if the DNA polymerase wasn't so good at its job, mutations would be much more common. So let's take a look at some of the types of mutations, and a lot of exam questions focus in on this kind of material. So let's take a look at the first type of mutation, which is called a point mutation. So in a point mutation, one base is exchanged for another. So here we're supposed to have CTGGAG, and then accidentally we've got CTGGGG. So where there was supposed to be an A, we put in a G accidentally. So this would have changed the DNA permanently. So let's take a look at how this one change in DNA could affect the potential protein that we're making from this DNA. So let's take a look. So the original DNA is ACA. So if you make messenger RNA from that, you would have the code UGU. And if you look up this codon in the codon dictionary, you would see that the amino acid that this codes for is cysteine. So in the normal healthy protein, we're supposed to have a cysteine amino acid at a certain place in our polypeptide. Now let's take a look at our mutation possibilities. So the first one, it was supposed to be ACA, and now we have changed it to ACG. So instead of an A here in the third base, we now have a G. So how is that going to affect the messenger RNA? We're now going to have a messenger RNA codon that says UGC. Now the good news is, if you look UGC up in the codon dictionary, it codes for cysteine. So it's the same amino acid. So even though the messenger RNA changed, we still got the right amino acid in the right place. So this would be called a silent mutation, because even though the DNA changed and even though we have a slight error there, we still ended up with the right amino acid and a fully functioning protein. So you wouldn't even notice that this had happened inside your cell. Now let's look at the next mutation, ACC. So we're supposed to have an A, and instead we have a C here as the third base. So how would that change the messenger RNA? Now our messenger RNA codon will be UGG. And UGG, if you look it up in the codon dictionary, codes for tryptophan. So tryptophan is a completely different amino acid to cysteine. So tryptophan would potentially make your protein, your polypeptide, fold incorrectly, give you the wrong shape of protein. So this is called a missense mutation. You're putting in a base, but it's the wrong base. It doesn't make sense. That's why it's called missense. So now when your protein folds, it might fold incorrectly, and then potentially you end up with a protein that either doesn't work or works better. But like I said, works better is very rare that actually happens. Most of the time, it doesn't work as well as the original protein. And then the third type of mutation here is we've switched the last letter to a T this time. So then when you switch the, create the messenger RNA from this message, you get UGA. And remember, this is one of our stop codons. So remember, the stop codon tells the messenger RNA or tells the ribosome when it's reached the end of the messenger RNA strand. So if you put in a stop codon where there's not supposed to be one, your protein or your polypeptide is going to be much shorter than it's supposed to be. And if it's much shorter than it's supposed to be, it's not going to work properly. So this is called a nonsense mutation, when you put in a stop codon and you end up with a very short polypeptide that probably does not work. So these are our different types of mutations. So remember, these are all point mutations. We're changing one base, a single base, and looking at what the outcome is going to be. So silent mutations, you changing a base, but it produces a synonym for the original codon. Remember when we looked at the codon dictionary, there was several codons that coded for the same amino acid. So sometimes if you just change one base in your trio of your codon, you don't change the amino acid. You still get the same amino acid in the same place, so you don't get any change in function. So those are our silent mutations. Now, your missense mutations are the ones that are really going to result in disease states, because in this case, you're putting in the wrong amino acid in the wrong place. So when your DNA mutated, it produces the messenger RNA that tells the ribosome to put in a different amino acid in that place. So again, the outcomes vary. It could have no effect. It could stop the peptide from working, or it could improve the function of the peptide. But most of the time, it's this one that it stops the peptide from working. Because if you change the amino acids, you change the folding. And remember, a lot of the functions of proteins are relying on that specific 3D shape. So that shape will be gone if it doesn't fold correctly. And then our last one, our nonsense mutation. This is where your change in your DNA has now produced a premature stop codon. So you're putting in a stop codon too early, so your peptide is much shorter than it's supposed to be. And in this situation, the peptide is almost always not functional. So these are the different kinds of base change mutations. Now, you can also get a mutation that's called a frame shift mutation. So let's see how that one works. And there's a great example of that down here. Even though it's a non-bio example, it just shows you how this frame shift would work. So when you read this first, the fat cat sat. This makes sense to us when we're reading. We're reading these words in threes, right? And with the letters we have, this is making sense, right? This is a sensical piece of writing. The fat cat sat, right? So when we read this in threes, it's making sense. But look at what happens if we take out one letter. So we remove that T there. So this would be deletion of a base in the DNA world. If you take out one letter, now does this make sense? Hef, atk, ats, at. No, so this now becomes nonsensical. So this is the same thing could happen with your DNA. You could insert an extra base or delete a base, and both of these are going to result in your codons being read wrongly. So you're going to end up with very different results, or sometimes nonsensical results. So again, the protein might still work, but it very often will lead to a dysfunctional protein. So it's called a frame shift mutation if you put in an extra base or delete a base, so that you change the reading frame and you change the order of the trios of codons that are being read. So that's the end of our video about mutations. So our last video is just going to talk about some of the ways that you can get a mutation or some of the causes of mutations.